Hey everyone, and welcome. If you've ever had a COVID test, then you've actually come face to face with the technology we're talking about today, real-time PCR. It's this incredibly powerful tool that lets scientists find, and I mean really find, the tiniest, most microscopic traces of genetic material. Honestly, it has completely changed the game for modern science.
So think about this for a second. The challenge is immense, right? You're a doctor, and you have to know for sure if your patient has a specific virus, like SARS-CoV-2. Or maybe you're a food safety inspector, and you're hunting for something nasty like salmonella. In both of those cases, the thing you're looking for is just impossibly small. We're talking a single strand of DNA in this giant, messy biological haystack. So how on earth do you find it?
To solve these kinds of high-stakes biological mysteries, scientists basically needed a molecular detective. They needed a tool that could not only pinpoint the culprit DNA, but also tell them exactly how much of it there was. And it had to do it all with incredible speed and accuracy. And that solution, that molecular detective we were all waiting for, turned out to be real-time PCR. It's this amazing method that can reliably find the absolute smallest traces of genetic material, pick out the exact sequence it's looking for, and then count it precisely, every single time. It was the detective we needed.
Okay, so what exactly is this technology, and why was it such a massive game changer? Let's dive in and break down the fundamentals. So at its core, real-time PCR, and you'll often hear it called qPCR for quantitative PCR, it does two really important things all at once. First, it uses the good old polymerase chain reaction, or PCR, to make millions and millions of copies of a specific piece of DNA. We call that amplification. But, and this is the real magic, as it's making those copies, it's also measuring how much new DNA is being created in real time, cycle by cycle. What's so cool about this slide is it really shows what a huge leap this was. Think about it. Conventional PCR was kind of like taking a photo, but only at the very end of a race. You'd know who finished, sure, but you had no idea how they ran the race. qPCR just changed everything. By watching the reaction as it happens, inside a totally sealed tube, the whole process got way faster, the risk of contamination just plummeted, and for the very first time, the results were truly mathematically quantitative. So how does it watch the reaction? How does it see what's happening? Well, the secret is fluorescence. qPCR cleverly translates the creation of new DNA into a light signal that literally gets brighter as more DNA copies are made. And this whole process gives us this beautiful signature graph for every single experiment. And here it is. This is that signature graph. We call it an amplification plot. Let's walk through it. See the bottom axis? That's the number of PCR cycles. Each one doubles the amount of DNA. Now the vertical axis measures the fluorescence, the light signal. And notice how for the first chunk of cycles, the signal's totally flat. That's just background noise, the baseline. Then all of a sudden, once enough DNA copies have been made, that curve just shoots upward exponentially. And that is the moment we've been waiting for. And here's the absolute key. Out of that whole beautiful curve, we really only need one single number. We draw a horizontal line, that's our threshold, just above all that background noise. The exact cycle number where our sample's curve crosses that line, that is its threshold cycle, or CT value. And believe it or not, that one little number tells us pretty much everything we need to know. Now, the absolute best way to get your head around the CT value is to think of it like a race, okay? So every sample is a runner. A sample that starts out with a lot of the DNA we're looking for gets a huge head start. A sample with only a tiny bit of that DNA, well, it starts way, way back. But they're all racing to cross the exact same finish line, that fluorescence threshold we just talked about. So obviously, the runner with the big head start is going to cross that finish line way earlier, in fewer cycles. And that's it. A lower CT value means a higher amount of starting DNA. It's so simple and so elegant. But wait, how was that light even generated in the first place? Well, there are two main ways to do it. The first way uses these simple dyes like cyber green that just glow whenever they stick to any double-stranded DNA. It works, it's simple, but it's not super specific. The other method uses these custom-designed probes like the famous TACMAN probe, which are engineered to light up only when the exact DNA sequence we're looking for is being copied. That gives you just incredible specificity. And this shows just how brilliant that TACMAN probe system is. So the probe itself is just a tiny piece of DNA. It's got a reporter dye on one end that wants to glow and a quencher dye on the other end that stops it. As long as they're close, no light.
But during PCR, as the little polymerase enzyme zips along copying the DNA, it runs right into our probe and just chews it up, breaking it apart. This separates the reporter from the quencher, and poof, it's free to glow. So more DNA copies means more broken probes, which means a brighter signal. So clever.
All right, so that's the core science. That's the how. Now let's zoom back out and see where this powerful molecular detective is actually being put to work, because its applications are just unbelievably broad and impactful.
I mean, just look at this list. The applications are really astounding. In medicine, it's a frontline tool for everything from monitoring how well a cancer treatment is working to measuring the amount of virus in an HIV patient. In public health, it's what keeps our food safe from nasty bacteria. It's used to diagnose diseases in our pets and livestock, and in research labs all over the world, it is the absolute workhorse for understanding the basic fundamentals of how our genes work. Yeah, with that combination of speed, sensitivity, and pinpoint accuracy, real-time PCR isn't just another gadget in the lab. For countless jobs, it has become the one and only, the definitive gold standard. And you don't have to take my word for it. This quote just says it all, doesn't it? Across so many different fields, real-time PCR is considered the gold standard. It gives us a level of reliability and accuracy that has made it an absolutely essential, indispensable part of modern science and medicine. So, let's wrap this up. If you only remember 3 things, make it these. Number 1, real-time PCR is our molecular detective that can find and count even the smallest, most elusive bits of DNA. Number 2, that CT value is everything. And just remember, a lower number means more of your target was there in the first place. And finally, number 3, this is not just some obscure lab technique, it is a cornerstone of modern science that is literally saving lives and protecting our health every single day. And that, of course, brings us to the future. I mean, with these new handheld portable devices coming out, the power of real-time PCR isn't stuck in big labs anymore. So the final question I want to leave you with is this. Where are we going to see these molecular detectives next? In your local doctor's office? Out on a farm field? In a remote village on the other side of the world? The story of this technology is definitely far from over.